We will get going here in just a couple of minutes. Good morning, everyone. Thanks for attending our first day of our plant pathology themed crop hour for 2025. Today, as you hopefully know, we're going to be focusing most specifically on white mold on soybeans. Before we get started, I just wanted to mention a couple of South Dakota specific updates. If you've heard me talk over the last couple of months, you may have heard me talk about SCN and that we were surveying last year. In an update on that is we've now found 12 additional SCN positive counties working toward the middle and western part of South Dakota. So just a reminder that soybean cyst nematode testing is free to South Dakota producers. So if you are in that center part of South Dakota and haven't ever checked for SCN, it might be a great time to consider doing that sometime in 2025. The other disease that I wanted to mention is Phytophthora root rot. We saw a lot of that in the wet fields in 2024. So if you believe that you had issues with that, please make sure that you take that into consideration as you make your management plans for 25. And with that being said, today our speaker is going to be Wade Webster from NDSU, and I will let him get going.
Awesome. Thanks, Maddie. So first off, I do want to kind of point out and I appreciate all of you guys, jackrabbits, letting a bison come in and talk about some white mold for you all. But I will preface this that I am actually a Montana State Bobcat and I'm not a true-blooded bison, so don't hold that against me. Okay, so my name is Wade Webster. I am the assistant professor and extension soybean pathologist up at NDSU in Fargo. So everything within my responsibilities is soybean diseases. And so Maddie reached out and asked me to talk specifically about white mold today. And so hopefully we'll kind of go through and learn some new things along the way about this disease. I'm sure many of you have seen this particular pathogen or disease before, but let's just get right into it. So many of us know that this is an important disease. We see this almost every year at some level. Some years it's at higher levels, some years it's at lower levels. But across the US or the North Central region every year, the extension pathologists, so Maddie and I and all of our colleagues come together and we estimate the different yield losses for the different diseases each year. And so if you can see here, okay, every single year except for 2018, white mold was above the top 5 most yield reducing diseases in soybean. Okay, that's a major deal because this disease, even though maybe some years we don't see it, it's a little drier, across the region, white mold is substantially reducing yields. Now this is a field dry land condition here. Okay, and we can see this is about 75 to 80% stand loss or yield loss because of white mold in some of these pockets. Okay, now it's not everywhere within this field, but there are pockets. And I'm sure many of you, if you've seen white mold before, have seen fields like this. But what exactly is white mold? And kind of understanding that it is a fungus called Sclerotinia sclerotiorum. Oftentimes we can call this Sclerotinia stem rot, Sclerotinia white mold, or just white mold as the disease goes. Now this pathogen, it is able to infect and cause disease in over 600 plant species. Most of these are broadleaves. So when we're thinking about this from a production standpoint, along with soybeans, we're thinking sunflowers, pulses, dry beans, and canola. Okay, so all of these are economically important crops that are also impacted by white mold. Now, white mold, as I said, very inconsistent, very dependent upon weather conditions. Some years it's very wet, we see a lot of white mold. In drier years, we may not see it as much, but it may still be present at some level. Now, the other important thing is that this white mold that survives is what we call sclerotia. And these sclerotia can survive for many, many years, up to about 10 years, especially in colder regions like in the northern Great Plains. Now, these sclerotia, okay, this is what they look like here, taken out of grain. This is a moderately infested field with white mold, and all of these little black structures are the sclerotia. This is the fungus, and when it goes into hibernation, okay, which means that those structures allow for this pathogen to survive for these very wet years, very dry years, and to continually produce new white mold mushrooms, or what we call apothecia. Now, some recent work that we've done shows that these sclerotia, the bigger and the healthier they are, they produce more apothecia and therefore more spores. The smaller the sclerotia, the fewer of these apothecia are formed, meaning the less disease risk in that next season. Now, these are what these apothecia look like. Okay, and these are small fruiting bodies, and you can see in this upper right-hand picture, that is the sclerotium body with apothecia growing out from it. So this is how these spores of white mold are released into that canopy of that crop. Now, these apothecia do not form just willy-nilly. They do require very specific environmental conditions. Okay, some of that being cooler temperatures, especially on that soil surface. Okay, between 50 and 75, usually 50 to 65 are kind of conducive, but we can get away with some warmer conditions. We also need some very high moisture. So this does not mean flooding. What this means is that consistent soil surface moisture. So if we're getting dew, if we're getting some light rainfalls consistently over a period of 1 to 2 weeks, these apothecia can start to form. And then we also need very specific light conditions. And these light conditions are oftentimes occurring when we get canopy closure. So when canopy closure is occurring, okay, that's when these apothecia are going to form. If we don't have canopy closure yet, these apothecia are not going to form. And this is very important when we're going to talk about management, especially with fungicides. Okay, so remember that point. Now, when we go on the field, okay, we may see apothecia on the left-hand side, but much more frequently, we're going to see these structures on the right-hand side. Okay, and the right-hand side are what we call bird's nest fungi. Now, these bird's nest fungi are what we call saprotrophs, and saprotrophs eat away at the plant material that's left on the ground. Oftentimes we'll see this on corn stover. And so you can see this here. There are two examples of corn stover with these saprotrophs or bird nest fungi growing on them. And one of the key diagnostics is inside of these little cups are the eggs. Again, looks like a bird's nest. Now, white mold, for example, these typically develop in clusters much smaller than bird's nest fungi, and they do not have these eggs inside of them. Okay, and you can see this picture on the bottom left is apothecia forming next to a dime, just showing how small these structures actually are within a field setting. Now, these apothecia, each individual mushroom itself can release up to 1 million spores. Now, 1 million spores is 1 million potential infections that could be occurring. Now, what they need is for a slight decrease in moisture tension. So what this could be is we have a layer of dew sitting over the top, say the middle of August or late July, and we get a gentle breeze going right over these apothecia. If that slight reduction in moisture tension occurs, then we see a puff of ascospores, kind of in this picture. And this is a really nice video here showing these puffs. Example. So that's a really great example. Again, just one apothecium releasing 1 million spores, okay, is a big puff. They're not continually being released, okay? They do exert a lot of energy to get those spores released. So it is small bursts. Now, once those ascospores are released, they need to get on flower tissue, okay? Specifically drying and dying flower tissue, okay? So when thinking about white mold risk and infection, we need these plants to be between the R1 and the R4 growth stage. And so it's just a quick reminder, and I'm sure many of you guys are on top of it, but within the soybean growth staging, R1 is when we have one open flower across that entire plant or on that main stem, okay? R2 is where we have at least one open flower on one of the upper twomost nodes. R3 is when these pods are just starting to form, okay, on one of the upper foremost nodes. And R4 is when these pods are about 3/4 of an inch long, right? So these flower structures are still going to be present up until through that R3, at least these drying down periods, drying down flower petals. And so this is kind of that risk window of infection occurring, okay, between that R1 to the end of that R3 growth stage. Now, once this infection occurs, okay, what's going to happen is that that fungus is going to infect and start to grow throughout that stem tissue, but it's not going to appear or come outward for 2 to 3 weeks, meaning that that infection is what we call latent. Now, after 2 to 3 weeks, we'll start to see this white, fluffy fungal growth, or what we call the white mold. We'll also see bleaching of that stem tissue, where that stem starts to turn that pale tannish coloration. And we'll also see new sclerotia forming on the outside of the stem. Now, these sclerotia are also going to form on the inside, but we are still in the middle of the growing season, so we haven't been able to identify that yet. Now, if this infection occurs at a high enough level or severe enough where we're girdling that whole main stem, we can see premature wilting occurring, which is what's happening here. So these plants are not dying because of the fungus, they are dying because they are not able to get water from the roots to the shoots, okay? Which is a really important part. The fungus is essentially suffocating and collapsing that vasculature within that plant tissue. Now, at the very end of the season, okay, we're going to see new sclerotial development forming again throughout the outside of that stem, the inside of that stem, as we can see here with that split soybean, and then also within the grain. And so there could also be sclerotia forming within the pods, replacing some of those beans. So these new sclerotia complete this disease cycle, being released back into the field as potential new inoculum for that next season or following 5 to 10 years. Okay. And so this is just, again, a very simple diagram of showing this whole disease cycle in which those sclerotia start in the soil. They move to the apothecia to release these bursts of ascospores, infect into that flower tissue, move up and down throughout that stem tissue, creating new sclerotia that are then released back into the field that next season. Okay. So how does white mold cause yield loss? And this is a really big question. And I love to show this picture here because it shows a lot, even though it just looks like a little bit of not so great soybean seeds. Okay. We see these sclerotial bodies. Now, these sclerotial bodies are what we call foreign objects. And if we were to take that to the elevator, there's going to be a pretty hefty discount for these if they have these foreign objects within these. On top of the sclerotia, we'll see reduced grain fill within these. And we can see so many of these individual seeds or beans that have not filled completely because white mold stopped that plant from developing or killed it off prematurely, preventing those beans from filling. And then lastly, we have these white chalky seeds. Now these are what we typically call mummies. And these mummies are infected with that Sclerotinia sclerotiorum, the fungus causing white mold. And what they do is they reduce the grain quality, they change the oil protein content of them. But also, if these are seed beans being planted the next year, these are going to potentially lead to greater infections outward from that or poor stand because of that. Okay, so again, within this one image, we have all these different examples of yield losses associated with white mold here. On top of that, we also, well, not on top of that, just as a summary here, okay, we have a lower seed weight, the sclerotia are present, these mummy seeds, again, the protein and oil are disrupted, and then potentially a lower germination rate if planted again the following season. So that's the biology, that's the disease. Now, thinking about it from a management perspective here, okay, how do we manage white mold effectively? Now, this is within a pathologist perspective. We like to show this diagram, and this is what we call the disease triangle. And as the disease triangle, we need all three parts of this to be present in order for disease development, okay? So we need a conducive environment. Again, we need consistent moisture, cool environment. We need the pathogen present. So Sclerotinia needs to be present in that field in order for white mold to develop. And we also need a susceptible host. Now, if all three of those are present at the same time, white mold will likely develop. Okay. Now, this is a little variation of that disease triangle, what we call the disease management triangle. And if you'll see off to the side of each of these three categories, we have different management practices. So in environment, we've got plant population, row spacing, weed control, and predictive modeling and rotation. So those are all mechanisms in which we are trying to manipulate that environment to make it less conducive for disease development. From the pathogen side, we've got pesticides, tillage again, and biological control. And then from the host side, or the susceptible genotype or cultivar or variety, okay, we can plant resistant cultivars. And then there's also differences between maturity group, which we'll talk about here in a little bit. So by incorporating those, we can start to combat white mold and manage it appropriately. So the first point management strategy that I'm going to talk about is crop rotation. Now, crop rotation is a really great tool for many diseases, but unfortunately for Sclerotinia, this pathogen can infect over 600 different plant species. And it's generally assumed that it's every single broadleaf plant that's out there. Okay, so that includes, again, canola, sunflower, dry beans. We can also have this on if we plant garden vegetables, but another very important part of this is weed management. Okay, many of these weeds that we have, the broadleaves, are all susceptible to Sclerotinia and can serve as an additional host to that in years in which we are either poor weed management, or if we are rotating to another host, okay, that's not a broadleaf, we still need to manage our weed populations. However, small grains and corn are both not hosts of Sclerotinia, meaning that they can effectively help to reduce and knock down some of our Sclerotinia populations. Now, the problem with this is that these sclerotia, as I mentioned before, survive up to 10 years in that soil. Okay, so if we're able to rotate out of soybeans for 5 to 10 years, which I know most of us are never going to be able to do, okay, we could potentially reduce that white mold pressure to low enough levels where it's not an issue. But these crop rotations alone are never going to help. But if we combine them with other strategies, they can be an effective tool to help reduce that pressure. So the next point is going to be cultural practices. And the three that we're going to talk about are seeding rate, row spacing, and then fertility management, specifically with nitrogen. So the first trials here, okay, this was large multi-state across 2017 and 2019 from Minnesota all the way over to Michigan. Now, across these locations, what we looked at is combinations of three different factors. And this included two different row spacings, so a 15 versus 30, which is our narrow and wide, four different seeding rates from 110,000 up to 200,000 seeds per acre, and then two different fungicide programs, which is a two-pass of approach, okay, or a predictive model-based program. These are compared against a non-treated fungicide treatment as well. So first, looking at white mold development on the left-hand side here, we're looking at is this disease severity index, or DIX. Now, this disease severity index is what we use consistently for measuring white mold development. And then on the bottom here, we're looking at the four seeding rates. So 110,000 seeds per acre on the left-hand side, all the way up to 200,000 seeds per acre. Now, what we found within these trials was that everything from 140,000 up had statistically similar levels of white mold development. And I'm sure many of you know this, but just as a refresher, okay, if these bars share the same letter, then they are statistically similar. They do not differ, okay, the same exact response. But if they have a different letter, then that means that they do differ. So again, the 110,000 seeds per acre reduced white mold development comparatively to some of these higher seeding rates. And the reason behind this, okay, is that the 110,000 seeds, what that does is it allows for fewer plants within that canopy, allowing for greater wind flow and greater air movement to dry out that canopy quicker. Now, if we have a more dense canopy, that moisture is going to be retained within that canopy for a longer period of time, leading to that sustained soil surface moisture. The next thing is looking at the combination here between the two row spacings on the bottom. Okay, so the 15 and then 30 inches. And then within each of those are the two fungicide programs in the non-treated. Now, the non-treated is this black bar with our predictive model in this dark blue, and then our light blue is our standard, which is an R1, R3 pass of approach fungicide. Now, same thing, we're looking at this DIX variable, and what we find is that across the board, 15-inch rows had higher white mold development than 30-inch rows. So that's important. Again, thinking about canopy closure, if we're having 15-inch rows, that canopy closure is going to occur about 10 to 14 days earlier than if we have 30-inch row spacings. And this is important because, again, these apothecia need these canopy closure to occur in order for them to form. Okay, so if we can delay that canopy closure, then these white mold apothecia or the white mold mushrooms are not going to form as quickly. On top of that, we do see an effect due to these fungicide programs. Okay, specifically, this standard has a reduced level of white mold development comparatively to the non-treated in both the 15 and the 30-inch rows. So we'll get into fungicides in a little bit, but understanding here, fungicides are an effective tool for reducing white mold levels. Okay, now white mold levels are great, but understanding yield is the next part, and this is also very important. So again, we're looking at 15 inches versus 30 inches on the very bottom, okay, on the x-axis. And then within each of those row spacings, now we're looking at each of the 4 seeding rates. So within the 15-inch row is where we see the highest yield at 170 and 200,000 seeds per acre. And that makes sense. Agronomically, this is how we optimize a lot of that photosynthetic capacity within these plants. Okay, we're covering all of that field surface soil so that we can maximize our yield. Okay, now as we go down to 110,000 seeds per acre within 15-inch rows, we see the lowest there. Now bumping over to the 30 inches, okay, that same response is present, but it's to a lower degree. Again, that 170 to 200,000 seeds per acre is still the highest yielding within 30 inches, but that 110,000 seeds per acre, that difference between the high and low is much reduced. Okay, now this is a caveat. Okay, 30-inch rows and low seeding rates can reduce white mold risk, but the yield is going to be reduced when we combine those together. And so this is one of our big takeaways within this particular trial, is that we recommend going to wide rows and low seeding rates in areas where white mold is an issue. Year in and year out, major issue, reducing yield. Now, in areas where white mold is not a problem, we do not recommend dropping seeding rates to around 110,000 seeds per acre or widening rows. Okay, so that is the caveat here within this particular study. Same thing with fungicides. Okay, looking at yield, the standard, which is again our 2-pass program, yielded higher than our non-treated. Again, this is not surprising, and we will get into more information on fungicides in a little bit. Now, this is the next important part, is the partial profit analysis, or the economics. So we accounted for the price of both the seed of the soybean at different seeding rates and the price of the fungicide programs that were applied. Now, from that, we looked at it under 3 different price points of the soybeans. So right now, what we're sitting at about a little over $10 a bushel, okay? We looked at $9, $12, and $15 per bushel to look at how feasible are some of these management practices here. Now, looking at the fungicide program, okay, these standard and the model. So when we applied a foliar fungicide at $9 a bushel, they did not pay for themselves in this particular study. Now, once we get to about $12 per bushel and above, then the standard and the model, so 2-pass and a model program, okay, yield or resulted in the same partial profit as the non-treated. Okay, indicating here that we can both have the same profitability and reduce the amount of white mold present within that field. Now, the greater risk of white mold, the higher white mold development within that field, these numbers will increase, more favoring those fungicides comparatively to the non-treated. The same thing, looking on the bottom here at the seeding rates, the 110,000 seeds per acre, again, was our lowest yielding, and we see this as being one of our lowest profitability treatments here as well. Now, what's very interesting is that this 200,000 seeds per acre, even though that is the highest yielding treatment, okay, we have the lowest profitability here as well. So what we recommend, again, these 140,000 consistently one of our highest profiting treatments within this particular study is where we want to be at in areas with low white mold risk here. Now, the next step here is looking at fertility. And this was another study done across the Midwest, multiple locations, again, from Iowa, Wisconsin, Michigan, and Indiana, looking at the combination of seeding rates, so two seeding rates at 100,000 and 160,000 seeds per acre, looking at three fungicide or what we call pesticide programs, because one is lactofen, which is actually an herbicide, okay, including our boscalid or Endura at R3 and boscalid at Forecaster, which is our predictive model. Now, the interesting thing that I'm going to focus on here is this nitrogen application. Now, the purpose of this is thinking about it more so from a manure perspective in areas in which we are working with dairies, that they are applying a lot of manure out prior to that corn crop, but going into that year two, if they're going back into soybeans, there could be a lot of nitrogen available in that soil for these particular crops. Now, what happens is when we have a greater amount of nitrogen present in that soil, then those plants are going to get bushier, they're going to get taller, canopy closure is going to occur sooner, all right? And we're going to see this increase in sclerotinia or white mold incidence. Now, this makes sense. The healthier the plants are, okay, the more conducive that microenvironment is going to be. And so by introducing in some outside nitrogen, in this case, it was urea, okay, applied at planting, we did see this response with higher white mold development comparatively to not applying additional nitrogen within a particular field. So understanding the role that fertility could have on sclerotinia or on white mold is very important. Now, the other thing here, in combination, looking at these two seeding rates in combination with the different fungicide and the lactofens, what I should call pesticide programs here, okay, we do see this response, okay. Now, if we have this high fertility down here, okay, if we can drop these planting populations down, then this will reduce that white mold risk. Again, showing what we saw before. Dropping those planting populations can help, especially in these high risk environments such as high soil nitrogen. Now, the next thing in here that we need to talk about is going to be variety resistance. And so genetic resistance is a very important tool when thinking about for white mold management. And one of the difficult parts of it is that there are not many options available. And so we did a large greenhouse study screening many different commercial varieties, and majority of those varieties were not able to stand up against sclerotinia. Okay. Now, that's the bad news, but the good news is that we do have some options available to us and more are coming out. And so this is work done out of my PhD when I was at the University of Wisconsin, where we bred three brand new, or three and an additional one that was already developed, public varieties that have been released for public growing that have high levels of white mold resistance. Okay. Now, everything here in these two figures over here demonstrate that white mold resistance, where we want to be similar to this 5282B. So we have the Sock, Marathon, and Rock varieties. These have all been released with similar resistance rankings to this 5282B. Now, these were actually run within the University of Wisconsin yield trials 2023, 2022, and 2024. And we can see over here on our yields, okay, well, I guess we'll go here to our 2-year average. Some of these do yield fairly well, particularly this 5282B, which is yielding about on average, okay, again, 2 different tests in 2023 at 74 bushels per acre. So on top of having high white mold resistance, it is also agronomically favorable soybean. And this is what these soybeans look like. Okay. They're very distinct in their phenotype. Okay. So this Marathon County on the left-hand side here does have a very black hilum, but it's a maturity of 1.5, and it is fairly decent bean. And we've grown it up here in North Dakota, and it still holds up fairly well. Sauk is another one with a maturity about 2.0. Same with Columbia, which is our 5282B. Okay. Sauk does have a clear hilum, where this Columbia does again have this black hilum as well. And then we also introduced and released this specialty bean, what we are calling Rock, which is our black seeded soybean with very high levels of white mold resistance. Now, on top of this, okay, this is just another confirmation of showing this 5282B as being a good soybean that has both high levels of white mold resistance up here, okay, showing this again on that DIX scale, comparatively to a very high susceptible soybean called Dwight. And then on the bottom, we're looking at yield. And across the board, 5282B yielded exactly the same as our Dwight, regardless of environment and treatments, indicating that you do not need to sacrifice yield when trying to go after high resistance within soybean varieties. Okay, it is possible, it is difficult, but it is possible to breed for this. And so there are becoming more options over the years. Now, genetic resistance, the problems here again, okay, very few highly resistant varieties. Some are able to fight it off. And this is a great picture here on the right-hand side of what we call the red node phenotype, where that infection had initially started right on that node. There was a flower tissue right here, okay, but the plant induced these chemicals within themselves to create this red toxic space, preventing that fungus from establishing and killing it off. Okay, so if you're walking around and you're scouting, this red node phenotype will indicate some sort of resistance present within that variety. Now, another thing that's important to think about is lower maturity groups. Now, this is primarily driven because of the flowers and the amount of time that those flowers are present on that soybean. If we have a lower maturity, those flowers are going to be there for a fewer amount of time than what we've had a higher maturity group. And so generally in some of those high white mold areas, we would recommend potentially dropping that maturity group if at all possible. Now, similar things, highly resistant varieties may not actually need a fungicide application, reducing the cost and the necessity for another input into a system if we can manage this with just resistance. Now, this is the next part that most of you probably want to be hearing about, and it's the chemical control. Okay, thinking about this from a fungicide perspective. Now, generally, we get a couple of questions, and there's a lot of questions for white mold, but these are kind of the big ones here. Okay, now the questions are: when should I spray? What droplet size should I use? How frequently should I be making fungicide applications? And what product do I use? Okay, all of these are great questions, and we're going to try to walk through each one of them here. Now, the first question is when to spray. The answer is very simple. Okay, it is between that R2 to R3. Okay, that's our best timing. Now, there's a caveat that we need canopy closure to occur. Okay, so if we are in R1 and canopy closure is occurring, then we need to make that application, but we need to do whatever is first. Okay. If we are outside of this flowering period, fungicide applications are not going to be effective. So if we are at this R5 growth stage and white mold is starting to show up, a rescue application is not going to be beneficial. So the first question or the second question on that list is, what about the droplet size? And so this is very important to walk through and think about it, because thinking about the micron size of those droplets and getting penetration into that canopy, even though we have canopy closure, is going to be very important. Okay. So if we're thinking about the amount of liquid within a 500 micron droplet, it is the exact same for one droplet at 500 as 8 droplets at 250 microns. And this is a better way of showing this here. Okay, so this was taken from Bob Wolf at Kansas State, these figures here. But you can see a 500 micron droplet at the top comparatively to 6,425 micron droplets and the amount of coverage that we get within that. Now, on top of that, the droplet size themselves will also impact the velocity or the speed in which these droplets are going into that canopy closure or going into that crop canopy. And so what we're seeing is we increase the droplet size over here to very coarse, again, thinking about getting close to that 500 microns, so 400 to 480, that velocity is increasing regardless of the pressure that these are being applied at, okay? Now, as we get finer droplets, so going down into fine and medium, this velocity is not as high. And so thinking about this again from a very dense canopy, those droplets going in at a lower velocity are not going to be able to get in through those leaves and get to those flower tissues to protect the plant. The other thing is thinking about drift. And I'm sure many of you have heard this from weed scientists as well around your area, but the greater or the lower the droplet size and diameter, the greater risk there is for drift. Now, the greater the size of that droplet, the lower the risk there is for drift. Now, these are some work done out of NDSU Carrington Research Station by Dr. Michael Wunsch, looking at the impact of spray droplet size. And this is a very complicated and intense figure here. So I'm going to try to break it up for you. So on the very top, we are looking at 6 different locations or 6 different varieties here, okay, listed across the top, all done in Carrington in 2019. And we're looking at the canopy closure of that crop. Okay, so this was measured these are all at canopy closure of what we consider almost 95% and above canopy closure. Next, we're looking at this white mold severity index again, across each of these different varieties, and we have a non-treated, we have fine droplets all the way up to a very coarse droplet size. Then we have soybean yield with the same treatments again, are non-treated from fine to very coarse treatments. Now, for the sake of today's talk, I'm just going to look at the combined analyses. So all of the different varieties combined for each of them. Now, looking at white mold levels here, okay, our non-treated had the highest amount of white mold, about 76%. Now that's very high levels. These are probably unrealistic levels of white mold development that we probably wouldn't see within a natural setting, okay? But what we're finding is that as we start to drop this down, okay, into very coarse, is that we're starting to see a trend towards lower white mold development. Same thing within yield, okay? The non-treated is yielding the lowest, and then our very coarse droplets are having the highest amount of yield. Again, protecting that crop when we're at canopy closure. So the next question here in consideration is fungicide application frequency. So again, taking work from Dr. Michael Wunsch out of Carrington, we're now looking at different maturity groups. So everything from double zeros at the top all the way over to a 1. And then across the bottom, we're looking at fungicide. So a no fungicide application, Endura 1-pass program, and Endura 2-pass program. Then looking at white mold, again, thinking about that white mold severity index. And then our yield here. And what we can find, okay, just looking across these maturities for white mold development is that white mold levels are highest in these higher maturities than they are in these lower maturities, again, because of that flower presence, okay, the timing of those. And we do see that in these lower maturities, okay, Endura 1-pass and 2-pass program had actually very little effect on yield. Now, if we take that all the way up to the upper end, okay, again, a 1 and a 1.3, which is a high maturity for this region, we do see this very positive effect. Okay, a 2-pass program of Endura, the first one being done at R2, okay, the second one being 10 to 14 days after that. We do see this effect positive benefit, okay, of 60 comparatively to 53 bushels protection of that crop because of an Enduro 2-pass program. Now, in addition to that, we've also done some we've done some uniform fungicide trials for white mold management across the Midwest with 5 different years looking at this data set here. Now, the first thing, okay, I want to point out is that yield here is on our gold bar. Our white mold disease severity index, or DIX, is again in our green bar. Okay, so our non-treated is on the right-hand side with this red arrow, and Endura 2-pass program R1 followed by R3 is over here on the left-hand side with the green arrow. Now we do see this positive trend where as disease development increases, yield decreases, and that's what we would expect. Now, across this trial, we looked at multiple different products, multiple different timings, and also multiple different application strategies, particularly some which we are calling our 360 undercover nozzle bodies. And these are drop nozzles into that canopy where we are making applications as a 360 degree to protect those flower tissues completely. Now, I'm going to break this up just a little bit here for you. So within each of these next 3 figures, I'm going to show on the left-hand side is this disease severity index. On the right-hand side is yield. Now, first we're looking at Endura or Boscalid. Okay, all of these are being applied at 8 ounces per acre. Now, our gold standard, our 2-pass program of Endura is in this dark blue consistently across these figures, and our non-treated is in blue on the right-hand side. Now, for white mold development, okay, we see the same levels of white mold control, white mold development for Endura applied at only R2, Endura following our Sporecaster predictive model, and then also Endura at our R3 time. Okay, now same looking at yield, we do see this clear yield protection or benefits, same levels between our Endura 2-pass program and our Endura only applied at R2. Now that yield starts to drop when we're looking at Sporecaster and our R3 apps here. The next thing we're looking at is Omega, and Omega is not a systemic fungicide, but a contact fungicide, meaning that it needs to get to that plant part to make that protection. And this is where these 360 undercover nozzle bodies came into play. Now, Omega, when we applied these 360 undercovers on the left-hand side here, we saw similar or low levels of white mold development comparatively to our Endura 2-pass program. But when we applied Omega over the top, okay, again, not getting to those flower tissues appropriately, even as a 2-pass program of Omega at R1 and R3, we don't see that level of control. And that's exemplified on the right-hand side here, again, with yield. The Omega 2-pass, or the Omega undercover, having similar levels of yield comparatively to our Endura 2-pass, but our Omega 2-pass program had the same yield as our non-treated. Now, the last one that I'm going to show here is a very common seed treatment, okay? Heads Up is oftentimes marketed for white mold control. The science behind it is that what they're trying to do, or this product is trying to do, is induce what's called systemic acquired resistance, in which these plants are triggered to, hey, start ramping up your defense system, and that this can protect against white mold control. Now, these are applied as a seed treatment as they are marketed. And across, again, all of these trials, we do not see the same benefit of disease protection comparatively to our Endura, and actually Heads Up has their same level of white mold control and yield as our non-treated here. So what are the next big things coming? Okay, and so from this, we're looking at some drone application work done at the University of Wisconsin, looking at white mold control, okay, under large plot field trials. Now, these are comparing the drone, again, using Endura at 8 ounces, but drones are applying at 2 gallons per acre, where a ground rig is at 15 gallons per acre. And this is a really great video showing these drone and the ground rig applications going on simultaneously. But the thing I want you to look at is the amount of disruption going on within that soybean canopy from this drone. just from those wings batting those canopy, we're getting better coverage into that canopy from those fungicide of the drone, okay? Now from this work, this is an image here, again, visual damage, okay, taken from drone imagery, looking at white mold levels. Now on the right-hand side, if you can see this, I really want you to focus on this bottom right-hand corner here. Now the first two plots here, this is our drone, okay, right here. This is our ground rig, and then this is our non-treated control. And we see that clear benefit here due to both the drone and the ground rig having similar levels here visually. But then when we look at this from a disease perspective from rating, okay, we see that the ground rig and drone had similar levels of disease incidence comparatively to non-treating at all. Similar response was seen with yield, in which we see the ground rig and the drone had similar levels of yield, again, with this benefit of protecting greater than our non-treated plots. So the next thing that we're going to talk about is predictive models. Predictive models, particularly, I've talked about these ad nauseum so far today in this talk, is a Sporecaster. And this tool is multiple, what we call logistic regression models. They're pulling in weather data from a cloud-based server for site-specific locations, and we're able to run different mathematical models from those environmental historical weather data. Now, this black figure here, this is what we call our daily risk value. Now, that moves up and down throughout the season, okay? And I put these blue arrows between the R1 and that R4 window. So again, that's our susceptibility window, okay? That's the only time when white mold infection can occur. Now, we have this other gold line in this figure that you see, and that's what we call our action threshold. And what that indicates is if that black line were to cross that gold line, that's when we would recommend a fungicide application. Now, in this particular field in Hartley, Iowa in 2019, that black line never crossed that gold line or the action threshold. Therefore, no fungicide application was necessary for management of white. Now, these models were integrated into a smartphone tool. These are all publicly available, completely free for you all to use and test out, okay, called Sporecaster. And what this does is this gives you a daily risk value for site-specific locations, and very, very happy with an accuracy of around 80% across all locations. Now, we're going to walk through this, and I'm going to show you an example of using Sporecaster here. So this is the app. We're going to open up and create a new field. Now at the bottom, we're going to click on the map, and we're going to go over to a field over in Marshfield, Wisconsin. This is an area where I know we have white mold consistently, and I'm going to drop that pin on that location, and I'm going to name this again Marshfield Field. Now from that, it's going to ask two different field variables, okay? The row spacing, 15 versus 30, or wide or narrow, and then is there irrigation present, yes or no? From that, we've created the field, and then you have to answer for that day, are flowers present? We know this is important, and we also know row closure is important. And so we have a visual diagram here showing over or under this threshold of canopy closure. So we're going to say, yes, it is over. And then we're going to look at July 15th of 2022. Okay, this is going to be, pretending like today is July 15th, and we're looking to see if we need a fungicide application. And that risk says it is low, meaning do not make a fungicide application. So we're going to back out and go to another field list. These are all the different locations that I have research trials at and that we're looking at. And we can see a map across the north central region. Okay, and these can be on your operations as well. And I'm going to go to our Oaks site. Now, Oaks, we're going to go same thing. Are flowers present? Yes. And row closure has occurred. And we're going to look at that same date again, okay? July 15th of 2022. And we're going to click Run Model. Now for this location, because the environment was different than Marshfield, okay, we're showing high risk, okay, bright red, saying high, anything over 10% should be applied, okay? So again, this is a great recommendation, a great tool to help guide making these applications. Now, the last thing I'm going to talk about here is biological control. And biologicals is a new area for white mold management of what we call biopesticides. Biopesticides through the EPA are broken up into three different categories of what we call biochemical, microbial, and plant incorporated protectants. And this is a really nice figure that I like to show, kind of separating these out. Okay, biochemical, what we would say things derived from different either plants, insects, different microbes themselves. So some of these could be the insect pheromones that we use in some of our insect traps. Plant incorporated protectants. Okay, an example of this would be like something genetically taken out that can be applied. Okay, the Bt toxin, which is used in corn. But most of what we're talking about for white mold and for pathogen protection is our microbial, which is our bacterial and fungal beneficial or biological control agents. There's also some viruses and protozoan species as well. Now, the activity and how do these actually work is also very important to understand. The first one is competition, in which two organisms are competing for that same space or resources. So imagining again, through white mold needs to form these apothecia, but if there's another fungus growing over top of that, preventing those apothecia from forming, just space competition, okay, then we could have reduction of white mold management or white mold development. Antibiosis, or essentially antimicrobial properties, in which one organism is putting out a chemical that is killing off the other organism. So these are very prevalent across our biological controls. Exclusion is preventing the pathogen from infecting. Okay. So if there was a pathogen that's capable of protecting and covering those flower tissues, being able to prevent those ascospores from causing that initial infection. And then this parasitism, which is the next one. And this one is most important for white mold. And I'll show a picture here in just a second of in which one organism is physically inserting itself, penetrating into the pathogen and consuming it, breaking it down. Now, the next one is what we call activation defenses. This is similar to that heads up product, the biology behind that, in which the organism or product itself is triggering the plant to increase defense system. Okay, saying ramp up your defenses, get ready to protect yourself from oncoming infections. And then there are a lot of RNAi technologies coming along, pipelines as well. And this is placement of specific genetic material or mRNA targeting different organisms to prevent infection through a lot of different genetic means. So the last thing here, and this is talking about that parasitism, is a fungus called Conotherium minitans. Now, Conotherium is oftentimes marketed as a product. I'm sure many of you have heard of this before or worked with it, called Contans. Contans is a fungus that is known to parasitize and degrade these sclerotia. And these are two, so this is not Sclerotinia sclerotiorum. This is a pathogen of peanut. It's a Sclerotinia species, but the concept is still the same here, okay? The one on the left is a very healthy sclerotia, okay? We can see that thing is very well intact. The one on the right-hand side is parasitized by this Conotherium. We can see all these thread-like materials are that Conotherium or Contans fungus growing all over that sclerotia. Now on the healthy one, okay, looking at the cell surface, all of those cells are sitting there. That's how the sclerotia protects itself, and it is able to survive for many years. But then when we get Contans involved, that protection is completely degraded. Okay, those sclerotia are no longer viable because this pathogen or this parasite has come in and degraded these sclerotia. Now, that was a lot to get through of white mold. Hopefully you all learned something of white mold management or the biology behind this. But I do want to mention, okay, that all of this work that has been done is not done by myself. This is done by so many different collaborators and partners and funding agencies to get all of these different locations and sites and all of this data kind of aggregated to present today. So thank you all for coming. And I think there's a few questions I see within the Zoom. So
Yeah, I can read those to you, Wade.
yeah, perfect.
First one is, did you consider 20-inch or 22-inch rows the same as the data in 15-inch rows?
Yep. So those were not directly evaluated. I understand being in Fargo, 22s is very common, especially within the sugar beet growing region. And so what we typically say is that anything above that 15 is what we call wide rows, anything below the 15. So if we're drilling our beans or if we're planting into 14, 15-inch rows, then that's what we call narrow. Now, that's one of my complaints, something that I want to get fixed in that app is not having 15-inch rows and 30-inch rows. So that is absolutely important for the biology perspective, but it all plays into the canopy closure. And so we know between the 15 and 30 that that canopy closure takes about 10 to 14 days longer for 30-inch rows. And so if you're kind of cutting that distance in half, you can probably cut the time in half too. So maybe an additional 5 to 7 days for canopy closure in 22 or 20-inch rows comparatively to 15.
All right, the next question is there any interaction between tillage and white mold?
That these are slides that I purposely did not present today because I didn't want to run short on time. But yes, there is an effect due to tillage. So tillage and tillage is kind of a broad concept. So one, I will say that if we plow, okay, such as mold borer plowing, all that's going to do is bury sclerotia deeper into the soil, but those sclerotia are not going to degrade. There's very little biological activity there. Now, if we're doing some light tillage or incorporation, if we can get those sclerotia into the soil where all of those microbes and microbial activity is occurring, those sclerotia will be degraded and break down quicker. Okay. Now, that being said, those sclerotia can still survive for multiple years. So, what we recommend, okay, is that if you can do a light tillage, get them incorporated, and then rotate out for 1 to 2 years out of soybeans or a white mold host, then that can help break those down even further. In no tillage, we don't see very little biological activity on the surface, and so these sclerotia just do not break down. Now, it takes, again, multiple years to see a clear benefit to this tillage. And so there are caveats, and my soil scientist colleagues do not like me telling you to go up and till your fields, but there could be some benefit, again, thinking about it from a microbial perspective.
All right, the next question is, did you evaluate any other fungicides other than Endura and Omega?
Yes, we did. So across that, we were looking at Delara Complete. We every year we run these UFTs. That was from that particular year. So between that year, I think we had in Delara Complete, Miravis Neo. This past year, we had Preaxor added in there. Um, we try to throw the gambit at it. We throw a lot of these different products, newer products that are coming out, so that we have the information to present and to publish on, make sure that you all are getting the best recommendations. So, if you would like any of those slides or data, please feel free to send me an email, and I will gladly share any of that information with you, so that you can make the most informed decisions possible for your operations.
All right, we got two more questions. We might go just a little bit past 11 if that's okay with you.
Oh yeah, of
The
course.
next question is, did you inoculate your trials or natural/field infestation?
Yeah, we do not inoculate any of our trials. Everything is natural infections. Now, what we do is we have a very strict rotation in a lot of our locations where we are either going between soybean and sunflower or soybean and dry beans to continually increase that white mold or new sclerotia production for that next soybean crop. So all of these are driven by those natural infections. Most of these locations also have irrigation to induce white mold development, but we do have some dry land environments also included within these trials.
All right, and then the last question I have on my screen is, The use of copper as a white mold fungicide hasn't been evaluated. Will we someday see evaluation of these less expensive fungicides?
Yeah, so we have actually evaluated some of these copper products. The copper products, there's a lot of hydrogen peroxide products that are coming out as some of our cheaper products as well. Um, they're very inconsistent in showing some benefit. Okay, they are not going to be as effective as what we might say Endura. Okay, um, because just the active ingredients themselves are not going to target white. Now, the problem here is that a lot of times some of these other individuals that are testing these products, if they're not getting the timing just right for these apothecia to form, then the data may not look good. But if we can get that application time just right, some of these cheaper products can still have a benefit of protecting those crops. So it's much more important for some of those products comparatively to say Enduro with a 14 to 21 day residual on it.
All right, we have one more question, but I'm just going to make an announcement since we are at 11. Thank you everyone for attending. If you have any more questions, wait, I got a few minutes so we can try to answer them. Please make sure you do our poll. Please make sure you answer the survey at the end of the week because we want to know if you like this type of content where we're focused more in on a specific topic. So please make sure you do all of that. And then the last question we got is how far can white mold spores move? If neighbor's field has a bad white mold infection, should I expect it to move into my field?
Yes, that's a huge question, and hopefully I can put that stress to bed. These spores do not move very far. They are pretty puny and weak. Once they get out of that crop canopy, the sunlight kills almost all of those spores. Now, the possibility is not zero that it can move from field to field, but that risk is very, very low. And typically what we see is about a 10-foot radius out from where that apothecia forms for new infections. So even if your neighbor does have white mold development, I would not go out and start spraying your entire field if you have not had a history. These sclerotial bodies are very much important for that disease. And if you don't have those sclerotia, that risk of white mold is still fairly low.
Okay, I think we've gotten through all of the questions. Thank you so much for taking the time to present today, Wade. Everyone, please feel free to reach out to him if you have additional white mold questions, or you can always reach out to me or Connie Strunk. And I think we'll go ahead and end. We do hope to see you on our wheat and corn crop hours, both tomorrow and Thursday. So with that, thank you all for attending.
Thank you.